Hello friends, welcome to a new video class. Today we are going to discuss the topic cellular respiration. We know that all living organisms in this universe need energy for performing their cellular functions. So the process of producing energy by the cell is known as cellular respiration. This cellular respiration may be of different types. Some cells produce energy with the help of or in presence of oxygen molecule. Then such type of respiration are known as aerobic respiration. For example, when we take the case of animals including humans, dog, you can see the aerobic respiration. And also in case of plants including algae, brio, pteridot, gymnosperm and angiosperm, you can also see the respiration, that is aerobic respiration. So we can see lot of example for aerobic respiration. Like that, some cells produce energy without the need of oxygen or in the absence of oxygen. Then such type of respirations are known as anaerobic respiration. Whereas when we move into the third case, some organism can able to perform aerobic respiration in presence of O2. Whereas they can perform anaerobic respiration in the absence of oxygen. So such type of living organisms are known as facultative anaerobes. So when we take the example, you can see few bacteria. Most of the bacteria you can see they can perform facultative anaerobic respiration. For example, Staphylococcus aureus, E. coli bacteria present in humans, like that Salmonella typhi bacteria, that those bacteria with that bacteria causes typhoid fever in humans, and the fungus Saccharomyces cerevisiae yeast and few mollusks. And also in humans you can see muscle cells, they can able to perform aerobic respiration in presence of oxygen, and they can also perform lactic acid fermentation. It's a type of anaerobic respiration. They can able to perform that anaerobic respiration during when we are performing when we are doing hard exercise. Then that during that condition, during that time period, the muscle cell perform anaerobic respiration. So the muscle cell also is an example for facultative anaerobe. So this is about the basics of cellular respiration. Next we need to know from which substrate this cell produces energy. So such kind of substrates are known as respiratory substrate. So that respiratory substrate or energy producing substances include, it may be either carbohydrate like glucose, fructose, lactose, arabinose, anything. Or also protein, lipids, fat, amino acids. All these substances are energy producing substances. So such type of substances are called respiratory substrate. Actually, during cellular respiration, chemical bond, especially carbon-carbon bond within this respiratory substrate undergoes cleavage. So, due to the breakdown or cleavage of chemical bond within this respiratory substrate, energy get released. That energy is utilized by this cellular organism to perform their function. That is the basics of cellular respiration. So, let's move into the broad concept of this cellular respiration. You can see cellular respiration mainly of two types, aerobic and anaerobic. Some perform both simultaneously, so that is why they are known as facultative anaerobes. And from this you can see, when we take the steps of cellular respiration, whether it may be aerobic or anaerobic, you can see the first step of all type of cellular respiration is known as glycolysis or EMP pathway. So you can see the representation of this glycolysis. In glycolysis, one glucose molecule, we know that glucose molecule is a 6-carbon compound. Its molecular formula is C6H12O6. And you can see the structure of that glucose molecule. Look at the cursor moving here. You can see the structure of glucose having 6 carbon. Carbon 1, 2, 3, 4, 5, 6. Here the numbering starts from top to bottom. You can see the sixth carbon atom, it says C6CH2OH. So here the 6 carbon glucose molecule partially, not completely, partially break down into 2 molecules of pyruvic acid. You can see the pyruvic acid. That is the basics of glycolysis or EMP pathway. Lysis means breakdown. Glucose is partially breakdown into 2 molecules of pyruvic acid. So you can see the glucose, you can see here glucose is a 6-carbon compound. So it partially breaks down into 2 molecules of pyruvic acid, and you can see the molecular formula of pyruvic acid here, CH3, C double bond O, COOH. So that pyruvic acid contains 3 carbon, whereas glucose contains 6 carbon. So one glucose molecule is partially break down into 2 molecules of pyruvic acid. So that is about the glycolysis. During that process, 2 molecules of ATP utilized, whereas we get 8 molecules of, whereas we get 9 molecules of ATP. So the net gain of glycolysis is total number of energy gained minus total number of energy utilized. That is 9 ATP minus 2 ATP. That is equal to 7 ATP only. We got final 7 ATP from glycolysis. So that is about glycolysis, and we'll discuss all the details of this glycolysis in next slide. Now let us move into the second steps of respiration. We are moving into the second step of respiration, especially the second step of anaerobic respiration. I already told you that the first step of aerobic and anaerobic respiration is common, that is glycolysis. Now let us move into the second step of anaerobic respiration, that is called fermentation. And before going to that process, we need to know the location of glycolysis. In glycolysis is always occurs within the cytoplasm of all cell, whether it is aerobic or whether it is anaerobic organism. In all type of organism, glycolysis take place within the cytoplasm only. So that is about glycolysis. So when we move into the second step of anaerobic respiration, that is fermentation. In fermentation, the 2 molecules of pyruvic acid either converted into lactic acid with the help of some enzyme. That enzyme is called lactate dehydrogenase, which we will discuss later. Lactate dehydrogenase. So that type of anaerobic respiration, second step of respiration, takes place within the human muscle cell. That is why when we are doing an excess work in one day, automatically next day we'll feel some pain in our joints. So that is due to the accumulation of lactic acid. And you can also see the lactobacillus bacteria that can able to convert glucose into lactic acid. That is called curd. We are very familiar about the curd. So that is due to the presence of this lactic acid fermentation. So in some other anaerobic organism, this pyruvic acid or pyruvate is converted into ethyl alcohol or ethanol. So such type of fermentation is called alcoholic fermentation, whereas the previous one is called lactic acid fermentation. During this process, energy get utilized. So all the details regarding with that type of energy production enzyme, all those things will be discussed later. So here we are only going to study the broad concept of this aerobic and anaerobic respiration only. Okay. So when we move into the aerobic respiration, so before going to that process, we need to know where is the location of that fermentation. So like glycolysis, fermentation also take place within the cytoplasm. So we can say that in anaerobic respiration, both glycolysis and fermentation takes place within the cytoplasm. But in glycolysis, they get a final energy of 7 ATP, whereas in fermentation, whether it is lactic acid or ethanolic or ethanol alcoholic fermentation, they lose their 5 ATP. They lost their 5 ATP. They lost energy equivalent to 5 ATP. So the final energy during anaerobic respiration is equal to 7 ATP from glycolysis minus 5 ATP from fermentation. That is equal to total energy of 2 ATP only. So when we move into aerobic respiration, its first step is also glycolysis. And we know that glycolysis is also known as EMP pathway because that was discovered by three scientists, E for Emden, M for Meyerhoff, P for Pernas. That is why it is called EMP pathway, Emden, Meyerhoff and Pernas pathway. So first step glycolysis takes place within the cytoplasm, and from the cytoplasm that newly formed 2-pyruvic acid from glucose then transported into mitochondrial matrix. So the second step is going to start from mitochondria. From that mitochondrial matrix, that 2-pyruvic acid is then converted into acetyl coenzyme A. Acetyl coenzyme A. By releasing carbon dioxide and producing 2 NADH2. And we know that 1 NADH2, that is nicotinamide adenine dinucleotide, one reduced NADH2 that is having H2. One NADH2 is exactly equal to the energy of 2.5 ATP, not is equal to 3. That is incorrect. And like that, one FADH2 is exactly equal to 1.5 ATP. It is not is equal to 2. And like that, 1 ATP is exactly equal to the energy of 1 GTP. This is the energy count of cellular respiration. So by using, we can say that during glycolysis, we'll get net energy of 7. Next, that process, that is the process of converting that pyruvic acid from the mitochondrial matrix into acetyl coenzyme is known as oxidative decarboxylation, because during that process, two carbon dioxide get released and one oxidation, that is NADH2, NAD plus 2H plus 2 electron gives rise to NADH2, that is oxidation. That is why it is called oxidative decarboxylation. So both the decarboxylation and oxidation takes place during the conversion of 2-pyruvic acid into 2-acetyl coenzyme. That is why that step is known as oxidative decarboxylation. That is also known as link reaction because that reaction connects the end product of glycolysis, that is pyruvic acid, with the next cycle, that is Krebs cycle. That is why this process is also known as link reaction. So this is about the link reaction which is taking place within the mitochondrial matrix. So the first step of aerobic respiration is glycolysis. In the under aerobic respiration, I'm not written glycolysis because it is common. So one is called glycolysis and second one is called oxidative decarboxylation or link reaction, which takes place in the matrix of mitochondria, whereas glycolysis takes place within the cytoplasm. Next, we have to study the third step of aerobic respiration, that is Krebs cycle, then fifth step of glycolysis. Fourth step of glycolysis, that is electron transport system. In case of oxidative decarboxylation or ring reaction, that takes place within the mitochondrial matrix, whereas the third step, Krebs cycle, also take place within the mitochondrial matrix, whereas the fourth step, electron transport system and oxidative phosphorylation, that process takes place within the mitochondrial inner membrane of mitochondria. Actually, during that process, ATP generated with the help of ATP synthase enzyme, in order to produce more number of ATP and accommodate more number of enzyme involved in electron transport system, inner membrane of mitochondria get folded into the form of crystal. That is about electron transport system and oxidative phosphorylation. All those details will be discussed during the successive slides. So let us move into the details of EMP pathway or glycolysis, which is the common first step of aerobic and anaerobic respiration. Let us move into that topic. Glycolysis or EMP pathway. It is also known as Emden, Meyerhof and Pernas pathway. Glycolysis consisting of 10 steps. So during that glycolysis, 7 ATP is the net energy gauge. So let us move into the each step. In first step, you can see the cursor. In first step, glucose, it is a glucose. It's a ring form of glucose. That glucose is a 6-carbon compound with a molecular formula of C6H12O6. That glucose, first of all, that glucose is converted into glucose 6-phosphate. And you can see the glucose 6-phosphate here. So what is the change here is that a phosphate group get attached at the 6th carbon, a phosphate group attached at the 6th carbon of glucose. So here you can see the red colored phosphate group that attaches on the 6th carbon. So the glucose then converted into glucose is converted into glucose 6-phosphate. So here we need an enzyme to attach the phosphate on the 6th carbon atom, on the carbon 6th carbon atom. So normally addition of phosphate is known as phosphorylation. The process of adding phosphate is called phosphorylation. That phosphorylating enzymes are commonly known as kinase enzyme. Here the enzyme is known as hexokinase because this kinase add phosphate group on the 6th carbon, 6th carbon. That is why it is called hexokinase. So in order to attach a phosphate on the 6th carbon atom, the enzyme need a phosphate source. The source of that phosphate is called the source of that phosphate is an ATP, adenosine triphosphate. That adenosine triphosphate contains 3-phosphate, triphosphate. So from that triphosphate, hexokinase take 1 phosphate and attach over the 6th carbon. That is why it is called hexokinase. So during the first step, the hexokinase utilize 1 ATP. So that is the first step. In second step, so before going to the second step, mostly this function is performed by hexokinase, whereas in liver and pancreas, this hexokinase gets replaced by another enzyme that is called glucokinase. So when we compare the hexokinase and glucokinase and hexokinase, they differ in their affinity towards glucose. Their affinity for glucose will be varied. So that will be discussed during the next slide. Now let us move into our second stage of glycolysis. During second step, that glucose 6-phosphate, you can see that glucose 6-phosphate then converted into fructose 6-phosphate. When you compare the carbon, hydrogen and oxygen and the phosphate number of both glucose and fructose 6-phosphate, in case of glucose 6-phosphate and fructose 6-phosphate, the number of carbon, hydrogen, oxygen and phosphate are same. That indicate that glucose 6-phosphate and fructose 6-phosphate are isomers. That is a compound having same molecular formula but different structure. And you know that glucose and fructose, they have same molecular formula 6H12O6, but their molecular structure is same, but their molecular but their molecular formula is same, but their molecular structure differ. That is why they are known as isomers. So here glucose 6-phosphate and fructose 6-phosphate are isomers. So during the second step, structural rearrangement or isomerization reaction take place. So that isomerization reaction takes place with the help of enzyme. That enzyme is called phosphoglucoisomerase. An enzyme which catalyzes isomerization reactions are commonly known as isomerase. So during the second step, glucose 6-phosphate converted into fructose 6-phosphate. That's all. When we move into the third step, that fructose 6-phosphate then converted into fructose 1,6-bisphosphate. What happened is that, what happened here? One extra phosphate get attached, get fixed on the first carbon atom, is fructose. Now the fructose become fructose 6-phosphate become fructose 1,6-bisphosphate. So here we need a phosphate to attach on the first carbon atom of fructose 6-phosphate. The source of that phosphate also from ATP, adenosine triphosphate. So here ATP cleaved with the help of water, that is hydrolytic cleavage or hydrolytic enzyme. Hydrolysis of ATP, here ATP get cleaved. One phosphate get cleaved from ATP with the help of water molecule by an enzyme. That enzyme is called phosphofructokinase, commonly known as PFK1. This is the main, this is an important step of glycolysis, that is the third step, PFK1, phosphofructokinase, that add one phosphate from ATP on the first carbon atom of fructose 6-phosphate. Now the fructose 6-phosphate becomes fructose 1,6-bisphosphate. And when we move to the fourth step, that fructose 6, so you can see the fructose 6-phosphate, you can see the fructose 6-phosphate here. The fructose 6-phosphate contains 6 carbon atoms. That fructose 6-phosphate break down into 2 3-carbon compound. One is called dihydroxyacetone phosphate and second one is called glyceraldehyde 3-phosphate. And when you look at the structure of dihydroxyacetone phosphate, that is DHAP, and glyceraldehyde 3-phosphate, both are isomers. Both contain 3 carbon atom and both contain same number of hydrogen atom, both contain same number of oxygen atoms, both contain same number of phosphate atom also. And you can see the structure of that dihydroxyacetone phosphate and glycerol 3-phosphate. That is exactly similar in the number of carbon, hydrogen, oxygen and phosphate group, phosphorus group, and they are also known as isomers, and they also interconvertible. They can able to convert, DHAP can able to convert into glyceraldehyde 3-phosphate. Like that, glyceraldehyde 3-phosphate can also converted into dihydroxyacetone phosphate, because when you look at the structure of, in the bottom region, you can see the structure of DHAP, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. Both are coming from glycerol, CH2OH, CHOH, CH2OH, glycerol. So here I am going to change this dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Suppose if you take this hydrogen atom, the hydrogen atom from the first carbon, look at the cursor from hydrogen atom. If you take this hydrogen atom and if you move this hydrogen atom into the second carbon atom here, and like that, if you move this hydrogen atom from OH into this region, now the structure became like this: CHO. This H and this H move here, so that is CHOH, and CH2O PO3 to minus. Like that, if you take this H of glyceraldehyde 3-phosphate and move to, and move to this hydrogen atom, and like that, if you move this hydrogen atom towards this oxygen atom, now the structure became like this: CHOH. Okay. So that is about the interconversion of this. It is very simple. That is, it is also a structural rearrangement isomerization reaction. Now, during the fourth step, fructose 1,6-bisphosphate break down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate with the help of an enzyme. That enzyme is known as aldolase. Especially we can call the fructose 1,6-bisphosphate aldolase. That is an enzyme which cleave into 2,3 carbon compound. Next, that is the fourth step. During the fifth step, this DHAP and glyceraldehyde 3-phosphate are interconvertible. That can able to move, they can able to convert with each other. So that conversion will be possible with the help of an enzyme called triose phosphate isomerase. Triose phosphate isomerase. That is the fifth step. So up to fifth step, the cell not producing energy, but the cell utilizes 2 ATP molecule. That is first and third step. And next sixth step, this glyceraldehyde 3-phosphate. We know that one glyceraldehyde 3-phosphate and one dihydroxyacetone phosphate are present. And during the next step, this glyceraldehyde 3-phosphate having 3 carbon atom, then converted into 1,3-bisphosphoglycerate. One phosphate get attached at the first carbon atom, but the source of this phosphate is not coming from ATP. That is why you can see the PI, not ATP. So during this step, one phosphate, inorganic phosphate, that is not from ATP, the inorganic phosphate get attached at the first carbon atom. Now it became 1,3-bisphosphoglycerate. But in order to convert this 1,3-bisphosphoglycerate, one hydrogen, that is one hydrogen molecule, that is not hydrogen, sorry, two hydrogen, that is two hydrogen atom, that is H2. H2 get released. That is, it is a reduction, a removal of oxygen or addition of hydrogen. It is an oxidation reaction. At the same time, that removed H2 used for the oxidation of used for the reduction of NADH2. You can see the H2 released from this glyceraldehyde 3-phosphate used for the reduction of NAD into NADH2. So here two reactions take place. One is called removal of hydrogen from glyceraldehyde 3-phosphate. Removal of hydrogen is called oxidation. And that hydrogen gets added to NAD, that is called reduction. So both oxidation and reduction take place here. So that is the sixth step, glyceraldehyde 3-phosphate converted into 1,3-bisphosphoglycerate. Here one molecule of glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. But actually two molecules of reaction take place because this dihydroxyacetone phosphate is free. That dihydroxyacetone phosphate also converted into glyceraldehyde 3-phosphate. Actually, one glyceraldehyde 3-phosphate is present here. And second glyceraldehyde 3-phosphate is formed from this dihydroxyacetone phosphate. So totally two glyceraldehyde 3-phosphate converted into two molecules of 1,3-bisphosphoglycerate. So 2 NADH2 get formed, 2 hydrogen get released, and 2 PI uses. So during the sixth step, we get 2 NADH2. I already told you that 1 NADH2 is equal to 2.5 ATP. So here we will get 2 NADH2, that is the energy equal to 5 ATP. And during the sixth step, now the sixth step is over. Now, seventh, eighth, ninth, and 10th step is pending. So when we move into the seventh step, 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate is a 3-carbon compound having 2 phosphate. One is at first carbon and one is at third carbon. That is 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate by removing one phosphate from the first carbon atom. So that phosphate released is utilized for the phosphorylation of ADP. That phosphate combined with ADP. ADP plus PI gives rise to ATP. So we'll get 2 ATP because we know that 2,1,3-bisphosphoglycerate is present and that converted into 3 phosphoglycerate. So 2 phosphate get released. That 2 phosphate combined with 2 ADP and form 2 ATP. So in seventh step, we'll get 2 ATP. So total ATP is equal to 5 plus 2 is equal to 7. So the seventh step of glycolysis. During seventh step, we'll get 2 ATP. That seventh step is catalyzed by an enzyme that is phosphoglycerokinase because it is a phosphorylation reaction. That is why it is called kinase. So here ADP phosphorylated into ATP. Next, this 3-phosphoglycerate. In 3, you can see the 3-phosphoglycerate. In 3-phosphoglycerate, the phosphate, the red colored phosphate present at the third carbon. So during that, from that state, 3-phosphoglycerate then converted into 2-phosphoglycerate. 2-phosphoglycerate. What happened is the phosphate from the third carbon shifted to the second carbon. So that became 2-phosphoglycerate. That is the eighth step. That step is catalyzed by an enzyme phosphoglyceromutase. It's a rotation reaction, mutase. So that is the eighth step. In ninth step, this 2-phosphoglycerate then converted into 2-phosphoenol pyruvate. What happened here is one H2 molecule, one H2 and one oxygen get released in the form of H2O. That is one water molecule get releases from this 2-phosphoglycerate. So not one water molecule. Actually, we know that after the fifth step, that is glyceraldehyde 3-phos. From this, the glyceraldehyde 3-phosphate is not one molecule because it is a two molecule. From glyceraldehyde 3-phosphate, all the reaction takes place by with the help of with the help of two molecules, that is 2-glyceraldehyde 3-phosphate, 2,1,3-phosphoglycerate, 2,3-phosphoglycerate, 2,2-phosphoglycerate. Every reaction occurs two times. So here 2-phosphoglycerate is converted into 2-phosphoenol pyruvate. So in one reaction, one water molecule releases. So here two molecules converted into two molecules of 2-phosphoenol pyruvate. So two water molecule get releases. Here in seventh step, 2 ATP produced. In eighth step, no ATP. In ninth step, no ATP. Only two water molecule get releases. That reaction, ninth step, that is 2-phosphoglycerate into 2-phosphoenol pyruvate reaction mediated with the help of enzyme. That enzyme is known as enolase that removes two water molecule from two molecules of 2-phosphoglycerate and will get two molecules of 2-phosphoenol pyruvate PEP. And that 2-phosphoenol pyruvate, that phosphoenol pyruvate, 2-phospho here, not phosphoenol pyruvate, actually it is a 2-phosphoenol pyruvate. The description is somewhat incorrect. Here, not 2-phosphoenol pyruvate, it is a 2-phosphoenol pyruvate because one phosphate group is present at the second carbon atom, 2-phosphoenol pyruvate. So this 2-phosphoenol pyruvate then converted into pyruvate. So here what happens is this phosphate molecule get released. So here two molecules of 2-phosphoenol pyruvate converted into pyruvate by releasing 2-phosphate. So that 2-phosphate get utilized by 2 ADP. So here the reaction is 2 ADP plus 2 released phosphate and form 2 ATP. So in the 10th step, we'll get 2 ATP. In the 7th step, we'll get 2 ATP. And in the 6th step, we'll get 2 NADH2. So the total energy produced is equal to 2 NADH2 plus 2 ATP plus 2 ATP. That is equal to 9 ATP. 2 NADH2 means 5 ATP plus 2 ATP plus 2 ATP, total 9 ATP. But in the first step and the third step, we will utilize one molecules of each ATP. That is, total energy is equal to 9 ATP minus 2 ATP, that is equal to 7 ATP. And one more thing is that the 10th step is catalyzed by an enzyme is called pyruvate kinase. That pyruvate kinase is also an important enzyme. And one more thing about this glycolysis is that some reactions are reversible reaction in case of glycolysis. That is step 2, 4, 6, 8, 9. 2, 4, 6, 8, 9 are reversible in glycolysis, whereas 1, 3, 7, 10 are irreversible reaction. 2, 4, 6, 8, 9 are reversible reaction, whereas other steps are irreversible reaction. So this is about the process of glycolysis take place in all kind of living organism, whether it is aerobic or anaerobic. Glycolysis step, that is the first step of respiration, is universal for all type of living organism, and that take place within the cytoplasm. Okay, that is about the glycolysis. Next, we need to study the regulation of glycolysis. The process of glycolysis is regulated in several regions that we'll be going to discuss here now. Next, let us move into the regulation of glycolysis. Glycolysis are regulated at some steps of the glycolysis. Among these, the first step. And we know that first step is the conversion of glucose into glucose 6-phosphate with the help of an enzyme, hexokinase, and in livers and pancreas, that hexokinase gets replaced by glucokinase. So the regulation of glycokinase, first regulation with the help of hexo and glucokinase. Actually, hexokinase is the enzyme which mediates the first step in normal cells. So that hexokinase shows high affinity, towards glucose for converting it to glucose 6-phosphate. Actually, that's a kinase, phosphorylating enzyme of glucose. So that shows high affinity for glucose than glucokinase. When we compare hexokinase and glucokinase shows low affinity, whereas hexokinase shows high affinity towards glucose. When the affinity is high, then their Km value or Michaelis constant is low. So hexokinase shows low Km value towards glucose, that is, they shows high affinity towards glucose. So that perform the phosphorylation of glucose to glucose 6-phosphate when the concentration of glucose, even though the concentration of glucose is very low within the cell, because that hexokinase shows high affinity towards glucose, they shows high affinity. So minimum, so if the concentration of glucose within the cell is low, then also it can able to perform the first step, that is glucose to glucose 6-phosphate. So that is an advantage in case of brain cell and RBC. Brain cell and RBC need energy, so they need more amount of energy, because we know that brain cell carry the main function of brain cell. The main function of RBC is to transport. They act as a the main function of RBC is that they can able to transport oxygen into all part of our body, all part of animal body by to carry oxygen. So they need more amount of energy. So during that condition, when the concentration of glucose is very low within the RBC, then also they need to produce energy by glycolysis. So for that purpose, hexokinase play a crucial role at low concentration of glucose. Like that, we know that brain cell produces energy only from two substrates. One is called glucose and second one is called ketone bodies. So whereas when we take the other cells in our body, they can able to produce energy from carbohydrate, glucose, protein, fat, fatty acid, everything. They can able to produce energy from different kinds of respiratory substrate, whereas brain cell can able to produce energy from glucose and ketone bodies. That ketone bodies also only in crucial condition only. So mainly they produce energy from glucose. So when the glucose concentration is very low within the brain cell, even though during that condition also the brain cell needed to produce energy, for that purpose hexokinase help a lot by producing energy by carrying out glycolysis even in low concentration of glucose within the brain cell. So that is a best mechanism. And one more thing is that if a cell produce more amount of energy by conducting more amount of glycolysis, then the cell gets saturated with energy. If the cell gets saturated, if the cell attains sufficient amount of energy, if the cell produces sufficient amount of energy, that is ATP, then the cell not need to carry out glycolysis glycolysis because they have a lot of energy. They already possess a lot of energy. So during that condition, they need to stop the glycolysis because they possess a lot of energy. So during that condition, the glucose 6-phosphate produced during the first step, the concentration of glucose 6-phosphate will be high within the cell because the rate of glycolysis will be high within the cell and they produces more energy. Now they need to stop the glycolysis. So that condition, that glucose 6-phosphate within the cell inhibit further glycolysis by inhibiting the hexokinase within the cell. So that process is called feedback inhibition. Feedback inhibition is if a substrate, if a product of a reaction inhibits its enzyme, then such type of inhibition is called feedback inhibition. Here glucose 6-phosphate inhibit hexokinase in order to prevent further glycolysis because the cell already possesses lot of ATP. So they don't need to produce energy by glycolysis again and again. So they stop. So they prevent the reaction of glycolysis. That is called feedback inhibition. That also takes place within the glycolysis, glycolytic regulation. And when we move into the glucokinase, glucokinase shows low affinity for glucose than hexokinase. So their Km value, Michaelis constant, will be high. For example, you can see the glucokinase in liver and pancreas. And we know that liver is the main organ which absorbs excess glucose from the blood and converted into glycogen. And like that, muscle cell also perform the same function. But here I am focusing only glucose. So in liver cell, glycolysis takes place, but the first step catalyzed by glucokinase. So glucokinase convert that glucose to glucose 6-phosphate. Then that glucose 6-phosphate move into the synthesis of glycogen pathway that is take place within the liver cell. So in liver cell, glucokinase is present. They shows low affinity because we know that liver cell this process happen when the liver cell contain plenty of glucose. That is I told you that when the blood glucose level is high, the liver cell intake all the excess glucose from the blood into the liver cell. So during that condition, the liver cell get filled with more amount of glucose. So during that condition, liver cell need only an enzyme which shows less affinity towards low affinity towards glucose because liver cell contain more amount of glucose. So they need low affinity enzyme to phosphorylate such kind of glucose because concentration of glucose is high. So the enzyme affinity, if the enzyme affinity is very poor, then also they can able to phosphorylate. That is the mechanism working behind the liver cell. And like that, beta cells of pancreas, we know that insulin is produced by the beta cells of pancreas. But how the beta cell recognize the blood glucose level get increases? Who informed the beta cells? So the answer is in this slide. You can see in beta cell also the glucokinase convert glucose to glucose 6-phosphate, but that glucose 6-phosphate then move into the glycolysis and converted into pyruvic acid. Thereby we'll get more number of ATP. That from each glucose we'll get net ATP of 7. And this newly created ATP by the activity of glycolysis started by glucokinase, and this more ATP then stimulate or trigger the production of trigger the production of insulin hormone within the beta cells. And that insulin hormone is a proteinaceous hormone consisting of alpha, beta, theta. That insulin again stimulate the glucokinase within the pancreas. So again and again, the process of glycolysis increases. The rate of glycolysis increases within the beta cells, and also that insulin move into the blood and control the blood glucose level by intaking the glucose into the liver cell and muscle cells. So here it is the mechanism which work behind the beta cells of pancreas. So that one also some kind of regulation is present. Next, second regulation is called PFK. I already told you that PFK means phosphofructokinase. It is a phosphokinase. It is a phosphorylating enzyme. And we already studied the third step of glycolysis. Fructose 6-phosphate is converted into fructose 1,6-bisphosphate. So before going to that detail, first of all we need to know phosphofructokinase is present in two different forms. One is called PFK1 and second one is called PFK2. Actually, PFK1 is directly involved in the third step of glycolysis, that is phosphofructokinase convert fructose 6-phosphate into fructose 1,6-bisphosphate. That is the third step of glycolysis. So during that process, we utilize one ATP also. And that fructose 6-phosphate gets regulated by two material. One is called ATP and citrate. Suppose if you imagine, if the cell contain more number of ATP, that indicate that cell perform more number of respiration and produce a sufficient amount of energy. So during that condition, the cell not need to produce more amount of energy further. So the cell need to stop glycolysis. For that purpose, if the cell contain more ATP, that indicate that cell attains sufficient energy. Then that ATP inhibits the third step of glycolysis enzyme, that is PFK1. Like that, citrate is the first product of Krebs cycle or citric acid cycle. Usually, glycolysis followed by Linger reaction, then followed by citric acid cycle, Krebs cycle. So, if the cell contain more amount of citrate, that indicate that that cell already performed glycolysis Linger reaction and produces more energy. So the citrate indicate that the cell contain more number of energy. Then also the glycolysis need to stop. So ATP and citrate prevent the PFK1, that is the enzyme of third step. Thereby we can stop the glycolysis. That is about, so that process of inhibition is called allosteric inhibition. Okay. And like that, if the cell contain more amount of AMP and fructose 2,6-bisphosphatase, fructose 2,6-bisphosphate. If the cell contain more number of AMP and fructose 2,6-bisphosphate, not bisphosphatase, bisphosphate, then that AMP and fructose 2,6-bisphosphate is an indication of cell cell shows a thirst toward energy. So during that condition, AMP and fructose 2,6-bisphosphate only, not phosphate, bisphosphate, act as an activator of PFK1. They stimulate PFK1 and they stimulate PFK1, thereby the cell carry out the third step of glycolysis faster than the normal glycolysis. So that one promote the glycolysis, and whereas the previous two one inhibit the glycolysis. Like that, PFK2, that is phosphofructokinase 2. It is an enzyme which converts fructose 6-phosphate into fructose 2,6-bisphosphate. It is not the step of glycolysis. It is another regulation. Fructose 6-phosphate is converted to fructose 2,6-bisphosphate. PFK2. And that PFK2 and fructose 2,6-bisphosphate are reciprocally regulated, and this fructose 2,6-bisphosphate again stimulate PFK1. I already told you that. And this PFK2 and fructose 2,6-bisphosphatase, an enzyme which can able to cleave fructose 2,6-bisphosphate, that is called fructose 2,6-bisphosphatase. Both are reciprocally regulated. So here comes the third enzyme, fructose 2,6-bisphosphatase, that are reciprocally regulated. Okay. The details of that fructose 2,6-bisphosphatase and PFK2 will be discussed now in detail. Okay, so let us study the PFK2, that is phosphofructokinase 2 group, and fructose 2,6-bisphosphatase, which are reciprocally regulated. First of all, you need to study the function, the reaction catalyzed by both these enzymes. PFK2 convert fructose 6-phosphate to fructose 2,6-bisphosphate, whereas fructose 2,6-bisphosphate is an enzyme which breaks down fructose 2,6-bisphosphate. So, let us study what happened here. When the glucose concentration is high within the cell, then PFK2 enzyme get dephosphorylated. One phosphate get removed from PFK2 enzyme. So dephosphorylation of PFK2 results the activation of that PFK2 enzyme. So if the PFK2 activated by dephosphorylation, then that PFK2 convert fructose 6-phosphate to fructose 2,6-bisphosphate. So the cell contain more number of fructose 2,6-bisphosphate. At the same time, dephosphorylation of fructose 2,6-bisphosphate, second enzyme, results in the inactivation of the enzyme. So PFK2 and fructose 2,6-bisphosphatase both get dephosphorylated, then PFK2 get activated and perform its chemical reaction, whereas fructose 2,6-bisphosphate get inactivated. Hence, the more number of fructose 2,6-bisphosphate produced within the cell during this condition activate PFK1, we already studied PFK1, lead to increased rate of glycolysis, leads to the increased rate of glycolysis. And also that increased rate of glycolysis due to the presence of fructose 2,6-bisphosphate produced by dephosphorylation of PFK2. That is happened here. Suppose if you imagine when glucose concentration is very low, then what happens is PFK2 and fructose 2,6-bisphosphate, both the enzyme get phosphorylated. Phosphorylation of PFK2 results in the inactivation, whereas phosphorylation of fructose 2,6-bisphosphate results in the activation. So activation leads to the cutting or breakdown of fructose 2,6-bisphosphate. Hence, fructose 2,6-bisphosphate concentration decreases within the cell. That leads to the slowdown of glycolysis, because if the fructose 2,6-bisphosphate concentration is very low, then PFK1 cannot be activated by an external agency like that of the previous case. So here glycolysis slow down. In the previous case glycolysis, the rate of glycolysis will be faster. That is the process happening. So here both are reciprocally regulated. PFK2 stimulate or activate glycolysis, whereas fructose 2,6-bisphosphatase, it slow down the glycolysis by inhibiting the PFK1 reaction by decreasing the concentration of fructose 2,6-bisphosphate. That is happening here. Next, let us move into the 10th end step of glycolysis, that is mediated by pyruvate kinase. And pyruvate kinase, I already told you that step 2, 6, 8, 9, that steps are reversible. But here, it is the 10th step of glycolysis, that is pyruvate kinase. It is an irreversible step. And this pyruvate kinase, it is also a kinase enzyme. I already told you that kinase enzyme that convert phosphoenol pyruvate to pyruvic acid, and that pyruvate kinase also regulated by an enzyme or a proteinaceous hormone. That hormone is called insulin. The insulin increases the activity of pyruvate kinase, because when the blood glucose level, when the blood glucose concentration is high, then the cell need to store or need to reduce that excess glucose. So during that condition, insulin increases the activity of pyruvate kinase, thereby the process of glycolysis, the rate of glycolysis will be increased. Whereas glucagon is an hormone which is produced by the beta cells of pancreas, sorry, the alpha cells of pancreas, and that glucagon is produced when the blood glucose level decreases than the normal level. So during that condition, glycolysis must be stopped, or the rate of glycolysis will be slowed down. We need to slow the rate of glycolysis. So during that condition, glucagon inhibit the 10th step of glycolysis, that is pyruvate kinase. Thereby, we can stop the reaction there. Normally, this pyruvate kinase is an enzyme that get activated in dephosphorylated form and get inactivated in phosphorylated form. When we add a phosphate group into the pyruvate kinase, then that enzyme get itself inactivates. If you remove that phosphate, that is phosphorylation, it becomes an active enzyme. Okay, so insulin and glucagon control the pyruvate kinase by phosphorylation and dephosphorylation mechanism. Okay, so let us try to understand the broad regulation mechanism in a tabular column. Here, first of all, you can see the enzyme hexokinase. Hexokinase get inhibited by one material, that is its product, that is substrate level inhibition, glucose 6-phosphate. Like that, the second one is called glucokinase, present in liver and pancreas, that get activated by the hormone insulin, whereas it get inhibited by glucagon. Like that, pyruvate kinase, the 10th step of glycolysis, that is that enzyme activated by an insulin, and second one fructose 1,6-bisphosphate. Like that, glucagon, ATP and AMP get inhibit the pyruvate kinase. Like that, the third enzyme of glycolysis, that is PFK, phosphofructokinase 1, that enzymes, activation of that enzyme mediated by insulin, adenosine monophosphate, fructose 6-phosphate and fructose 2,6-bisphosphate. Like that, glucagon, ATP, citrate, low pH, that is acidic pH, and cyclic AMP inhibit the action of PFK1 also. This is the regulation of glycolysis. So here we are going to stop. We can study the further step of aerobic and anaerobic respiration during the next class. Thank you to all.